Hello, my name is Grant Reeves, and for my master's thesis research project, I am investigating how defective Filopodia elongation disrupts central nervous system development. And so, the general paradigm of my research begins with the generation of a random mouse mutation using ENU mutagenesis. And that random mouse mutation will then be corresponded to a specific phenotype that I'm looking for, which generally pertains to nervous system development or nervous system dysfunction. And then we will take that phenotype and we will map it to a specific genetic location on a chromosome and then determine which gene corresponds to that phenotype. And so my specific project was started through the generation of the NMF418 homozygous mutation through ENU chemical mutagenesis. And these mice exhibit severe hind limb hypotonia. As you can see here, this mouse indicated by the red arrow has very low tone in its hind limbs and it cannot move particularly well. And these mice rarely survive to adulthood. Only about 4% of the mice we would expect to live to adulthood actually survive. And so what are the phenotypes that these mice display that we originally found? And the first of these phenotypes is a failure of the vertebral column to close around the spinal cord. So we can see on the wild type here on the left that the vertebral column is completely fused, leaving no opening or exposure of the spinal cord. Although on the right here in the NMF418 homozygous mutants, there is an opening in the vertebral column. The bones have failed to fuse, resulting in an exposure of the spinal cord to the external environment, as is indicated by the red bracket. The second of these phenotypes is an elongation of the cerebellum, as is similar to Chiari malformation. So on the left here in the wild type, there is a proper foliation of the cerebellum as indicated in Roman numerals from 1 to 10. We can see that they are ordered relatively well and do not have any elongation along the rostrocaudal axis. But in the NMF418 homozygous mutant mice, we see that there is elongation along the rostrocaudal axis, and that the foliation in the cerebellum is not as ordered as it would be in the wild type mice. The third of these phenotypes is an expansion of the lateral ventricles, as is consistent with hydrocephalus in humans. So LV indicates the lateral ventricles, CC indicates the corpus callosum, as these are coronal sections going through the center of the brain like this. And, we can see that on the wild types, the lateral ventricles are quite small, and in the 418 homozygous mutants, the lateral ventricles have expanded to a significant extent. So these three phenotypes in my mice mirror spina bifida with the failure of the vertebral column to close, Chiari malformation with the extension of with the elongation of the cerebellum, and hydrocephalus with the expansion of the lateral ventricles. And so what gene is mutated that causes these defects? We identified FMNL2 as a candidate gene for the NMF418 homozygous mutation. And we localized it to a 4 megabase pair region on chromosome 2, and using next generation whole exome sequencing, we identified a single nucleotide polymorphism in the gene FMNL2. And we identified that single nucleotide polymorphism in FMNL2 as A to C mutation in a splice acceptor site between intron 9 and exon 10, and this results in the improper excision of exon 10 from the FMNL2 transcript. And this is indicated through gel electrophoresis, where we can see on the left here, there is a wild type band that has a length of 270 nucleotides, and on the right here there is a homozygous mutant band, and that has a length of 195 nucleotides, and that corresponds to the length of exon 10 that is being improperly spliced out. And so this improper excision of exon 10 results in a greatly reduced protein level for the NMF418 homozygous mutant mice. We can see that in the cortex, cerebellum, hippocampus, and even in non-neuronal tissue of the lungs, there is a greatly decreased protein level. Through Western blot analysis, we can see very thick, dark bands for all the wild type samples in these tissues, whereas in the homozygous mutant bands, there is homozygous mutant wells, there is no, essentially no protein. And so what function does FMNL2 have in the cell? FMNL2 is a cytoskeletal organizer protein, meaning that it polymerizes together microfilaments and extends these things called Filopodia. So these little hair-like projections that you see in these human melanoma cells are called Filopodia. And FMNL2 acts together with proteins called IRTKS, also CDC42, to polymerize together F-actin chains that allow these hair-like projections to come off of these cells. And this is believed to be very important in cell migration. And so we think that the defects in FMNL2 lead to defective cell migration in the 418 homozygous mutants. And so we have a hypothesized this is a hypothesized mechanism for the formation of Chiari malformation in humans, as is proposed by McClone in 1989. And this all begins with a failure of the neural tube to close, as is consistent with spina bifida in humans. And that leads to disrupted cerebral spinal fluid pressure dynamics that lead to a formation of a small posterior fossa. So the posterior fossa is a bone that sits at the back of your head, right back here, and surrounds the opening in your that allows your brainstem down into the spinal cord. That is called the foramen magnum, which is this big hole indicated here. And that smaller posterior fossa leads to a herniation of the cerebellum into the foramen magnum because of restricted size. And that herniation of the cerebellum into the foramen magnum disrupts cerebral spinal fluid pressure dynamics even further and leads to expansion of the lateral ventricles, as is seen in hydrocephalus. And so we have seen previously that in my mice, they have spina bifida, so we believe that this is due to an initial failure of the neural tube to close. We have seen that there is an elongation of the cerebellum, as is consistent with Chiari malformation, and we have seen that there is an expansion of the lateral ventricles, as is consistent with hydrocephalus. So I decided to look and see if there is this restricted posterior fossa content. And to do this, I did cerebellar sagittal sections of postnatal day 0 mice. So these are neonatal mice. And I found that in wild type mice, there is quite a bit of space surrounding the cerebellum between the exterior bone, which is the posterior fossa, and the cerebellum. Although in the 418 homozygous mutants, we see that there is very, very little space, if none, between the cerebellum and the posterior fossa. And we, in fact, see that there is herniation of the cerebellum into the foramen or past the foramen magnum in the 418 homozygous mutants, as is by definition Chiari malformation. We can see here there's a little division between the bone and the cartilage of the foramen magnum, and the cerebellum is indeed extending past that. So we found that there is indeed restriction of the posterior fossa content in my mice, resulting in this herniation of the cerebellum into the foramen magnum. So next I wanted to investigate if there was this failure of the neural tube to close originally that results in this downstream cascade. So to do this, I'm doing transverse sections of E11.5 embryos. I am taking mice, monitoring when they get pregnant, and then 11.5 days later, I am taking down the mother, euthanizing her, and collecting the embryos, and then embedding the embryos in wax and cutting through them essentially horizontally through the embryo. And I am showing you a section through the embryo at about the middle of the embryo. So the structure that we're going to be concerned with is called the neural tube. This is what this will look like in a transverse section. And we can see that the neural tube is located on the dorsal surface of the whole embryo here on the left, and that the neural tube is properly closed in wild-type mice, and there is no opening on the dorsal surface. Now, the interesting part is that when we look at the 418 homozygous mutant mice, there is also no opening on the dorsal surface, which appears to run contradictory to what we would have thought previously. But there is clearly no failure of the neural tube to close, even in the 418 homozygous mutant mice. So We have seen that there's a restriction of the posterior fossa content. We have seen that there's herniation of the cerebellum into the foramen magnum. And we have seen that there is expansion of the lateral ventricles in my mice, but we found that the catalyst that is supposed to cause this downstream cascade of phenotypes is inconsistent with my findings. And there is no failure of the neural tube to close that is originally supposed to cause all of this. So there must be something that is actually disrupting cerebral spinal fluid pressure dynamics, and that is believed to be defective bone progenitor. This hypothetically could be defective bone progenitor migration, as we see that there is a defective patterning of the dorsal column in these mice, as is indicated by the failure of the vertebral column to close and exposure of the spinal cord. And this could somehow disrupt cerebral spinal fluid pressure dynamics. And so I am in the future going to look into how hypothetical bone progenitor cells are migrating to the dorsal surface of the embryo, and how this could potentially be disrupted. So I would like to thank the Ackerman Lab. I would like to thank Dr. Susan Ackerman, my PI, and Joyce Tran, my direct mentor. I would also like to thank the Edwards Yeckel Fellowship for Science Communication for supporting me in these endeavors, and of course, my friends and family. I really appreciate it. Thank you very much.